here the shape of this binding site is triangular, right? So it will say no, you can nicely sit on me. Make sense? Because you are square, I am triangular. Make sense? It will go here, it will say no because I am round. It will go here and this will nicely accept, fit here. Make sense? Because shape matches. This is called key lock. You put the right key in right lock, then it will start, right? The engine of your car. When you put the right key, turn right, it will start the car. So when that fit will be perfect, then the signal will go to inside the cell from outside, okay? And intercellular machineries will start to work, okay? So that is how the receptors, integral proteins, work as receptors. If you take this medicine, this round one, it will not fit here, not fit here, but it will nicely fit here, and signal will go inside. Okay? So that is how some integral proteins act as receptors, and that's why, you know, this is not very simple. When the medicine companies, you know, pharmaceuticals, they produce the medicines, molecules, they must study what kind of receptors are present in what kind of tissue, make sense? What kind of cells. If they want to make a medicine that will work on the liver tissue, liver cells, they must know the shape of the receptors, right, in those cells. Otherwise, the molecules will go there but will not be able to work. Does it make sense to you? So, the molecules must match to the receptors to work on it. And that's why different medicine works on different organs. Not all medicines should work on everything. Make sense? So, different hormones work on different organs, structure. Some integral proteins work as enzymes, and you know enzymes convert one chemical to another. That's what they have shown. They have shown these two integral proteins are working as two different types of enzymes. That's why they are converting the chemicals from one form to another. Okay, so those are the functions of the integral proteins. Now we'll see the functions of the peripheral proteins. I already mentioned, wrote down on the board. These two are the functions. Intercellular or cellular junctions or joinings, same thing, and cell-to-cell recognition. Okay, I wrote here already. I think you have already. Okay. Cellular junctions or joinings and cell-to-cell recognition. Now, cellular junctions or intercellular joinings, very simple. This is one cell, this is another cell, okay? And two cells are attached by those junctions. Make sense? So, holding the cells together by attaching the cell membranes. So, that is intercellular junction. So, this is one cell, this is another cell, and they are attached to each other by cellular junction. Peripheral protein. What is cell-to-cell recognition? This is very important. You know, all of you know that if I take tissue to graft, for example, skin, if I take from your own body and put from one place to another, your body will accept it. Make sense? You all know that, right? But if I take from another person and try to put on your body, the chance of rejection is very high, right? You know that. Tissue rejection, cell rejection. Why that happens? Very simple, because your cells of your body, of your tissue, knows which cells are from your own body, which are coming from another body. Make sense? It recognizes. So, that is the cell-to-cell recognition, and that is helped by the peripheral proteins. So, some peripheral proteins help to recognize another cell, which is a very important function of the the proteins, peripheral proteins. You know that when microorganisms, those are cells too, bacteria, right? Try to invade into your body cells should be immediately recognize that, right? It is not from your own body, coming from outside. Okay? And then immune system will work, right? To destroy. So cell-to-cell recognition, also tissue transplantation, organ transplantation, cell, transplantation, all are factors here. So, what are the common types of junctions? Cell-to-cell, intercellular junctions. 3 intercellular junctions are very common: tight junctions, desmosomes, and gap junctions. Tight junctions, name is telling you, very tight. Almost no chemicals can pass through the tight junction. Desmosomes. Desmosomes form attachment by forming the protein structures like this. Look at me. This is one cell membrane. From one cell membrane, a structure comes out, protein structure comes out like this, another next cell membrane like this, and they hold each other like this. Okay, very strong connection, attachment, difficult to separate. It's like zipper in your clothes. You use the zipper, and you know how strong is that. So that is desmosome. And gap junctions have many holes or pores through it. Its name is telling you gap. So lot of pores or holes or channels through the gap junction. That's why the chemicals and molecules can easily move from one cell to next. Now, in some tissues in your body have plenty of desmosomes. Example, in your cardiac muscle, heart muscle, you have lot of desmosomes. So the heart muscle cells are strongly attached to each other, which is very important for the heart, because all muscle fibers must work simultaneously at the same time. Make sense? If just think that if you guys are standing here side by side and holding your hands very loosely, then if something strongly pushes you, you will get dispersed, separated, right? But if you hold your hands very strongly, right? Now if something pushes you very strongly, all of you will move in the same direction. Make sense? So in the heart, the muscle fibers are held together by desmosomes, so they work together. That is one thing. Also, in the heart muscle, you have plenty of gap junctions. Why? Because of gap junctions, the chemicals can quickly pass from one cell to another. So it's like open doors. So they can communicate, the heart muscle fibers can communicate to each other quickly. Because this is one heart muscle fiber, this is next, this is next. 2 types of junctions. Desmosomes hold them very strongly, and gap junctions have channels. So the fluid or liquid can easily move from one to another chemicals. So they can talk to each other quickly. It's like open door between them. And these are the desmosomes. So all of them should contract at the same time. Both are important. Okay, here you see 3 cells are side by side, and they have shown the junctions. The first is tight junction. You see here, this is how the tight junction attach the cell membranes. Now if you see the structure of a tight junction, this is the structure below. It's like, a very simple way to think is, this is one cell membrane, this is another cell membrane, they are attached to each other and then stitched together, okay, by proteins. They are stitched together. So two ships are stitched together. That is tight junction. Desmosome, I mentioned that desmosome is like this. So protein structures, they hold like zipper. Here in the bottom you see the structure of a desmosome. What happens in desmosome? This is a cell membrane, this is another cell membrane. In the inner side of the cell membrane you have plaques. Okay? And then from the plaques the protein structures arise. Those I showed you like this, and they get attached to each other like this one from this side and another one from that side, or like this, okay? So like this. A very strong connection. So these structures are called linker glycoproteins. You see there, or cadherins. Remember this name, I may ask you. So linker glycoproteins arise from the plaques and get attached to each other like zipper. Okay, cadherins or linker glycoproteins. So this is a very strong intercellular attachment. Gap junction, you see channels are present or pores are present in the gap junction. So the chemicals can easily move from one cell to another. So cells can talk to each other quickly. If they are closed, one cell cannot talk to next. So chemical exchange quickly occurs through the gap junctions. Okay. So make sense? Okay, now transport across the cell membrane. Cell membrane is a semi-permeable membrane. I have mentioned already. That means partially permeable. It does not allow everything pass through it. Some chemicals can pass through it. So selectively permeable. Yes, selectively permeable. Only those chemicals are needed for the cell to be able to enter, and those chemicals should get out, will be able to get out, not everything. Okay. Now, through the cell membrane, two types of transport occur. One is called active, another is called passive. Which one needs ATP? Active. Makes sense? Active, because it is saying active. So active transport requires ATP. That means energy is required for active transport. Makes sense? Passive transport does not utilize ATP or energy. It is like, this is a hill. You try to climb up, okay? You want to go up to the hill. So to go up, you need what? Make sense? So that is active. Now if you want to slide down, you just sit there, right? You can go down. So active and passive. Make sense? One requires ATP, another does not. Now you tell me, this is you, okay, and your child is sitting on your shoulder. your child is sitting on your shoulder. Now you are climbing up, okay? So who is active, who is passive? You are active, right? And your child is going up, but no, he is not using any of his energy, right? He is passive, right? So sometimes some molecules sit on another molecule, and that molecule actively moves, but the molecule sitting on it is passively going. Okay, so active transport occurs only in living cell members, because ATP is only produced by metabolism, right? You remember? And metabolism only occurs in living body, right? Living cells. In dead body, dead cells, no ATP, right? No metabolism. So active only in living cell members, but passive can occur in both living and non-living. Now, passive could be three types: simple diffusion, facilitated diffusion, and osmosis. So this is a cell membrane or plasma membrane or whatever, selectively permeable membrane, and in this side you have glucose solution. These are glucose molecules in water. So you have glucose and it is dissolved in water. Okay. This side of the membrane you also have glucose solution. That means you have glucose and water. Okay. Concentration in this side, the concentration of solution is high. In this side, it is low. So, this membrane is semi-permeable, so it will allow the chemicals to pass through it. Now, in this condition, remember, the solutes, very simple, think very simple. Who is more in this side? Solute is more, right? In this side, glucose is less, right? Number of glucose molecule is less because this is low concentration. This is high concentration, right? So, to bring equilibrium, the goal is, if you know the goal, it will be very simple. The goal is what? Bringing equilibrium, same concentration in both sides. That is the goal. So now if you know that, then you can easily tell who should move from this side to this side, because this side glucose is less, right? So glucose will move from this side to this side. Make sense? So movement of glucose from high concentration to what? Low concentration. That is called simple diffusion. Simple diffusion. Movement, now let's go back. Movement of solutes, because solvent is water always. Movement of solute, which is glucose in this case, from high to low. That is simple diffusion. Make sense? Movement of solute. Now you tell me, who is more in this side, less in this side? Water, right? Water is less in this side. So water will move from this side to that side to bring equilibrium. Water will move from this side to this side to bring the concentration down. Solid will move from that side to this side to move the concentration up. Make sense? To bring equilibrium. So that movement of water from low to high, remember always it will be from what? Low to high. That is called what? Osmosis. Okay, so those are two passive, and another is carrier mediated or channel mediated. You have already seen, I have explained, this is a channel, right? So I told you those molecules have a smaller size than the size of the channel, will be able to pass through it, get in or out, right? So, helped by the channels. And carrier, I also mentioned that if you sit on someone's shoulder, right, and then move from one place to another, you are not utilizing energy. Make sense? So, carrier is taking you from one place to another. Make sense? Carrier is taking you. Like this one, example. The carrier is taking you. So, that is also passive because no energy is used. So, carrier mediated or channel mediated. This is a test, very simple. You can do that. You take a U-shaped tube and put a semi-permeable membrane in between two solutions. In the right side, you see in left picture, right side of the tube, you see high concentration. In the left side, you see low concentration. So, in this case, equilibrium, to bring the equilibrium, the solute will move from the right to the left and water will move from left to the right. So, one is simple diffusion, another is osmosis. And in the right side, you see in both sides the concentration got back to normal. Okay. Same. Now, if the semi-permeable membrane, in this case, you see, has very tiny holes, only allow the water to pass, not the solid. So, in this case, what will happen? Only water will pass from low to high. Only osmosis will occur. Make sense? Because solute cannot pass. So, in the right side, you see, since the water has moved from left to right, in the right side, the volume increased, but the goal is not the volume. The goal is what? Goal is what? Equilibrium of what? Concentration, right? Not the volume. So, volume could be different, but goal is equilibrium of concentration, same concentration. Okay. Now, we'll see the response of a living cell in three different types of solution. This is a solution. Okay. The concentration of this solution, for example, 10 milliosmol. That's the unit of concentration. Okay. So, this solution has 10 milliosmol concentration. If you put a cell, living cell, that has semi-permeable membrane. Okay. So, this is the cell. Okay. You drop it inside. Now, inside the cell, the concentration is, for example, 5 milliosmol. So, concentration is high in outside, right, and low inside. Now, you tell me, just I have explained here, which way the water will move, from low to high or high to low? Water will, which way the solute will move? High to low, right? And water will move in opposite direction, from low to high, to bring equilibrium, right? So, always remember, water will move from low to high. So, now you tell me, concentration is high here, low here. And water will move which way? From always? Low to high, right? Make sense? Water will always move low to high. So, which way the water will move? Look at this way, everybody. It doesn't have, you have already though. So, concentration is low here, right? And high here. And water, the rule is, water will always move from low to high. So, water will move this way, get out. So, the shape of the cell will get what? Smaller, shrink. Make sense? Now, if I change the solution here, concentration is 2 milliosmol now. Water will move from low to high. That means water will get in, right? Because low is now outside. 2, inside is 5, right? So water will move this way. So the cell will get what? Bigger. And may even rupture, right? If more and more water gets in, will get big and may even rupture. Now if the concentration is same both inside and outside, then no change because water will not move, right, in any direction. So the shape of the cell will remain what? Same. Remain the same. Make sense? So no change. So when it is 10 milliosmol compared to inside the cell, outside is hyper. So we'll say this solution is hypertonic. When it is 2 compared to inside, this is low. So we'll say hypotonic solution. Hyper. When it is same inside and outside, it's called isotonic. Okay? So those are 3 conditions, and the cell will respond differently in different conditions. Okay? So here, if you go over, but the rule is always water will move from low to high, and that is called the osmosis. Make sense? That is called osmosis. Okay. I will take your attendance, then